Welcome everybody. Today we have Jared Schuetter for our series. Jared Schuetter is a principal data scientist with over 15 years of experience tackling complex and innovative challenges that demand the integration of statistical methods and machine learning AI techniques. Jared received his PhD from The Ohio State University, his master's from Bowling Green State University, and his BS, both those were in statistics, and his BS in mathematics and education from Denison University. Throughout his career, Jared has contributed to diverse and impactful projects such as threat prediction and signal processing for DNA sequences, genomic forensic analysis, classification of chemical and biological spectral signatures, and image processing in various domains. Jared, like me, is at Battelle, and I've had the pleasure of working with Jared several times in the past. We did forensic genomics work together, developing methods to extract CODIS STR information, which is standardly obtained another way, not from DNA sequencing information. And Jared was a huge contributor to that project. So it's been a pleasure to work with Jared for a long time here at Battelle. He joined in 2009 and has been a great researcher all of that time. Today, we're going to hear a little bit more about Jared's work doing classification of chemical and biological spectral signatures. And so I will turn it over to Jared.
Great. Thank you, Nancy. And thanks to the NISS and all of you for attending. So I gonna walk through some, well, let me go back. We'll talk through today about this machine learning for airborne biological hazard detection, and just to give you kind of an overview of what's in today's talk. So I'll start off just with a brief background about me and Battelle, if you're not familiar with Battelle, and also the domain of airborne biological hazard detection, in case you don't work in that area. Then I'm going to spend most of the time today talking about this system called the REBS, which is a device that we've developed here at Battelle on research and development dollars primarily, and it's used for biological hazard detection. So we'll talk about that. Most of what I wanted to share was about like how that system works and what kind of data it produces, and the ways that we've analyzed it. And then along with that are like all the problems that we had to solve to figure out how to analyze the data. You know, something fun about working in industry is things can get kind of messy, especially when you're in kind of R&D mode. So hopefully you'll find some of that interesting. And then I'll end with a summary of, you know, maybe some of the lessons that I've learned from working on that project and others at Battelle. Okay, so first we'll just do some background here to catch you up on all the things I'll be talking about today. So Nancy kind of said all this in my bio, but you know, I have a background in statistics actually, but since I've been at Battelle, it's been mostly machine learning and more recently some deep learning in a lot of different areas across the company. So a lot of the biological and chemical detection work I did was on the REBS, which I'll talk about here soon. And then also in oil and gas, human forensics, and a lot of biology. More recently, you know, I've stopped doing as much technical, like hands-on technical work myself and moved into some task leadership and business development and strategy. Here on the right is a picture of what our division at Battelle looked like back in 2009, and here's me when I was a lot younger and I hadn't really dealt with real data sets yet. Still had hope. I guess I still have hope now, but a little more realism in my mindset. So okay, so that's me. Now we'll talk a little bit about you know, biological threat monitoring or aerosol detection, really. So the goal that we want to tackle here is we want to be able to detect any kind of biological threats that might you know, come into an environment. There are a lot of different places where you want to make sure that the things people are breathing in are safe, you know, and in a couple of different areas. So first, in terms of just sort of like a national security mindset, you might want to protect U.S. citizens, in certain settings, places where people tend to congregate. So like a football stadium or a train station or a park, like I showed here, just an example from overhead. There could be military applications. So, the armed forces, if they were to be attacked by some kind of biological weapon, you might want to detect that at like a forward operating base. And then also for like primarily health medical type applications, you may want to detect any kind of biological contaminants. So, in pharmaceuticals, or if you're a company developing, say, you know, tubes and things that will go into medical devices, those need to be developed and built in an environment that's clean. And so, you may want to be putting some devices in a clean room to monitor to make sure there aren't any, mold spores or fungal spores floating around, or in a water supply, say. So just using this park as an example, the idea is you're going to place some sensing devices of some sort throughout that environment. And, ideally, these would be like passive sensors. So any kind of particles floating around the air, when they come into contact with one of these devices, the device will in some way, try to detect and maybe identify what that particle is. And then by tracking that information over time, or by using the results of an identification kind of algorithm, you can figure out whether there are any anomalies. Is there like a biological material in that environment that shouldn't be there, or are the levels of biological particles going higher or lower than you would expect, which might be indicative of some kind of an event. And so if something is detected, then you would send an alert to some kind of end users, maybe first responders, or some kind of dispatch, something like that, to try to get somebody to look at the situation. So, in this picture on the right, we have maybe there's some kind of plume of biological material here. Hopefully you can see my mouse. Some kind of plume of biological material that's like emitted from around the center here and flowing to the southwest. And maybe like two of these sensors kind of detect particles that are anomalous, that are associated with that plume. This would be like an alert that would be sent to someone to say, hey, there's something happening here. So as you can imagine, there are lots of different considerations in a problem like this. Like, how many sensors do you need to have to protect against certain types of scenarios? How complex do they need to be? So do they need to be just simple detectors, or do they also need to identify the material? How quickly do they, like, what's the time resolution on their readings? How quickly can they analyze the particles, those kind of things? What are all the different potential scenarios you might want to detect? And then also, given a particular sensor configuration, how do you test and evaluate how robust that network would be in response to lots of different events that might happen? Okay. So I can pause there briefly, but if there aren't any questions, I can keep going.
If anyone
Okay.
has any questions, you can put them in the Q&A. Thank you.
Thanks. Okay. So I'm trying to keep this high level so I don't lose anybody. So hopefully you're still with me at this point. Okay, so the area Battelle's been doing a lot of research for decade and a half, I guess, at this point, is in the design. Well, in part, what we do is we've tried to build our own sensor solution for these types of problems, but and that's the REBS, which is here under Raman spectroscopy. I guess actually, there you go. But there are some other options out there. So just to give you a quick overview of the sorts of sensors that people have developed, a lot of the sensors that are used in this field are these the light-induced fluorescence sensors on the left. These tend to be like really low-cost sensors, so you can put a lot of them out in the environment. They're lightweight and kind of easy to use. The drawback to these types of instruments is that typically they're just detection based. So they're just looking at the fluorescence of these particles in different bands of light to try to identify whether they're biological, and then kind of counting them. And so all you'll get from these devices is you'll be able to see whether there's an increase in the volume of biological particles, the concentration of those particles over time. But you're not necessarily going to be able to identify what those particles are. And you can imagine in like springtime, you're going to see an influx of biological particles just because you're going to have like pollens and other things that are being released from all the plants that are growing. And, if you're not accounting for that, you could have a lot of false alarms from systems like these. But anyway, those are used a lot. There are other devices using spectroscopic methods, whether it's mass spectrometry or Raman spectroscopy here, that try to go a step further by actually collecting a signal from the particle and trying to identify what the biological material is based on that signature. And so I'll be talking about REBS today. REBS is the selling point, I guess, for REBS is that unlike at least some of the older mass spectrometry type methods, the REBS doesn't really need reagents or anything. It kind of just like works autonomously over a long period of time. The only real issues are with batteries and with like a tape reel that I'll talk about later that needs to be changed like every so often. But in general, like those instruments can kind of run on their own for a long period of time. Other sorts of instruments need, if you're using like gas chromatography with the mass spec, you might need reagents and things to allow that process to happen, and you're going to need much more frequent maintenance. So the REBS was kind of a solution to use fewer resources and need less maintenance over time. But aside from those kind of spectroscopic things, there's some other methods, newer things that are being pioneered now, focused on more like sequencing analysis. So, microfluidic sensors that you can pair with a PCR, or you can extract a sample and take it to a lab to do assays or sequencing. I think people are trying to figure out ways to use like Oxford nanopore technology to do faster sequencing, like with the MinION, if you've heard of that. And then there are other methods using like remote sensing or microscopy. Some of these things require some more human interaction. So, and I see a question here too. Yeah, so in terms of particle size, yeah, these instruments tend to include filters and things as well to kind of at least get rid of the larger particles, or to measure the sizes of the particles. There are only certain ranges of particle sizes that are going to be breathable and of danger to people. So, and that's a feature of the REBS, which I'll talk about in a minute too. Okay, so just a quick background on Battelle, if you're not familiar. We've been around since 1925. It was originally, Battelle was founded in the will and trust of Gordon Battelle, who was primarily worked in metallurgy. So a lot of Battelle's early work was focused on materials and those kinds of solutions. Over time, we've kind of grown to work in all kinds of different areas, which on the right, you can see we kind of bucketed them into three big areas: national security, health, and environment and infrastructure. But Battelle itself is it stands on these three pillars, which are on the left here. So there's a whole contract research part of Battelle, which is where I work. And then because we're a nonprofit independent research lab, we have a strong charitable giving portion of our business, which we focus a lot of those on STEM education. So building STEM schools across the country to help students build those critical thinking skills in those areas. And then the third piece of Battelle is we actually do laboratory management of 9 of the national labs around the country. And so that's another big piece of the business that's kind of separate from contract research. And this particular talk about airborne biological detection, that's all in this national security space primarily that's been done. So quick overview of what Battelle does in terms of SBIRNI, which is chemical, biological, radiological, nuclear, and explosive materials. So any bad things, basically, whether they explode or irradiate you or infect you. We have over 500 scientists, engineers, and technicians. One of the things that's unique about Battelle is we have the largest privately owned biosafety level 3 facility in the US. So, the different biological agents come with different biosafety levels. And, typically by the time you're at biosafety level 3, we're talking about things like anthrax or, ricin or plague. And so we can work with those materials and do like test and evaluation of devices for, at that level, whereas a lot of other labs outside the government can't do that. We have a pretty large business testing different medical countermeasures, a lot of it animal testing, but we've been moving in the direction of in vitro testing, using like organ on a chip sort of technology. And we also have high containment capacity and throughput. So we support a lot of like FDA approval of different medical countermeasures, vaccines, things like that. And then we, in general, have capabilities across the board from like early stage characterization of threats, analysis of like sensor data to detect threats. We do test and evaluation of devices. We build our own devices. And then we also help with the operations planning and training, with first responders or military or what have you. Okay, and then just a quick note on the test and evaluation, because I think it might be relevant later. This picture on the left, actually, that is a REBS down here in this, it's called an HVADS chamber, but we kind of have different scales of testing that we can do for devices. So these small chambers on the left, you would like introduce a material here on the top and it flows through this tube or whatever, this rectangular kind of container here. And the material gets sampled through this intake down into the device where we try to detect and identify what it is. But then we also have like room scale kind of chamber testing that we can do where you can release a material into an entire chamber and have multiple devices try to detect it. We've got kind of large scale facilities for handling live agents, especially those BSL-3 agents I was talking about. And then we even have some large scale outdoor facilities where you can kind of have a more naturally occurring outdoor environment and blow some kind of material down a tunnel and see if devices can detect it. Of course, you're limited in what kind of danger, like you can't send too dangerous stuff through those facilities, but that's also a useful tool that we have. All right, so just some quick overview of the REBS, which is the thing producing all the data I'm going to be talking about. So we started developing REBS back in 2006, and this kind of just shows the evolution of like where we've been moving. I'm not going to go through all these, but I mean, in general, the trend is that the early models we just kind of built to try to get this whole process working. At the time, people had done Raman spectroscopy in a lab to characterize materials, but hadn't really tried it in this context. So a lot of the early iterations were just trying to get it to collect good data and allow us to like build the algorithms to try to detect and identify these things. And then as we started trying to mature the product and harden it for different applications, primarily for like more military or national security focused applications, a lot of uh, the requirements of the field started to creep in. So it was like, okay, if we're going to actually use this thing, at a forward operating base, say, we need this device to be light enough to where, two people can carry it. And it needs to be ruggedized to where if they drop it on the ground by accident, it's not going to break the thing. It needs to operate in high heat or extreme cold. It can't have like dust getting in and gumming everything up. So a lot of these later versions were really like trying to shrink the size, add a bunch of these kind of ruggedized features, make it easier to maintain. And of course, all of those things, when you shrink a device and you're trying to add all these bells and whistles on it, that means basically you've got less space to collect good data. So it was kind of like competing forces, I guess, all throughout the development. We want to make the device easier to sell to people and more useful, but also, if there's less space for the innards that actually produce the data that you're getting, or you're trying to detect things faster because that's what end users need. Now you got to like update your algorithms and things to be able to handle maybe noisier data or to make earlier decisions on less information. So there's always kind of a challenge there, but in general, you can kind of see that trend. And so, our current generation is called this Rebs Plus, which I'm not going to talk about. Most of what I'm going to talk about is older Rebs stuff. But yeah, the Rebs Plus we built for like urban biothreat environments. So it's a sensor that can be placed on mobile, uh, on a mobile vehicle, like say a police cruiser or something, driving around a city to help identify, where things might be happening in an urban environment. But we've also tried to tailor reps for indoor environments as well, like hospitals and things like that. Okay, so that's like my brain dump about everything to do with the field, and so hopefully I still haven't lost you. Big picture is we're trying to detect biological particles in the air. We're trying to identify what they are, and we built a device to do that. That's the Cliff's Notes. So now I'm going to talk to you a little bit about the data that we produce on this device and how we used to analyze it. It's matured a little bit since then, but I'll share what I can here. Okay, so my background is statistics, not physics or biology, so so I'll do my best though to explain how this stuff works. So if you're not familiar with Raman spectroscopy, basically you have some kind of like a molecule. I'm showing here polystyrene, which is not biological, but it's just an organic kind of polymer molecule. We actually use this to test the REBs to make sure it's working. We just throw some polystyrene beads in it basically and see if we detect it. So the idea is you're going to shoot a laser, it's going to hit the particle, and depending on the different chemical bonds and things that are in the particle, there's going to be some scattering of that laser. So the laser is at a specific wavelength of light, and when it scatters off this particle, you're going to get some different scattering at shorter wavelengths than what the laser light is, and then you're going to have some scattered light at longer wavelengths. And so we're interested in the longer wavelengths here. And so the idea is you're going to look at all the different photons at these different longer wavelengths and sort of bin them by the wavelength. And then if you plot them on a plot here, this wave number, which is the horizontal axis, that's it's essentially the shift in the wavelength from the original laser wavelength. So, you know, some of these shifts will be at larger wavelengths than others, but by counting all the photons that you get at each of those different shifts, you'll get this spectrum. And so, so from a single laser hit of the particle, you get this entire vector of data, basically. And these intensities are going to be counts. So, you know, in general, you're collecting photon hits on like a CCD array. So they're going to be like pixels along the array, and that's going to bin the photon counts into different ranges of these wavelength shifts. So that's how Raman works. And so with the REBs, basically most of the machinery in the REBs is just getting the particles underneath a laser so that you can shoot them with the laser and measure the spectrum. That's the idea. And so, you know, the first thing is we've got this kind of smokestack thing on the top of the REBs. It's going to pull particles in from the environment and send them down a tube here. Anything that's outside of the range of respirable particles. So, you know, like most of the stuff we're interested in is 2 microns or 2 micrometers in diameter. Those are like the sizes of cells of a lot of biological organisms or like spores and things. Anything that's too large gets filtered out through a filter that we have in place. And so everything that comes through is going to be pretty small. These things get, we basically apply like a static charge to them, and then that makes them stick to a tape. And so down here you can see like 2 spools, or yeah, like 2 spools, I guess, down here on the left and right. And there's like a metal tape that goes up and through this instrument. It's kind of like an old school cassette tape. And so these particles will stick to that tape. It's like a aluminum coated mylar tape. And so they stick to that with a static charge. And then what we do is we like scroll the tape to move it from here over underneath this laser. This whole thing is the optical system on the right. And so that's where we shoot the laser and hit the particle. We also collect all the scattered light in the CCD array, which is in a spectrometer. And that gives us a spectrum. Well, kind of. It gives us the data that will eventually be turned into a spectrum. So first, just to give you a sense of what's happening, this is like what a spot looks like on the tape. So after those particles get the charge and they get deposited on the tape, this is a picture that we took at one time under a microscope of what that looks like. So you can see the different particles that are stuck on here. And then this optical unit here, it kind of has these components to it. So there's the laser, a spectrometer, and a camera. So this laser is going, the way that we were doing analysis at this time is we're kind of hopping this laser across the spot. And so you would like start the laser at some spot over here on the left side, and then we do these little jumps, and we kind of see like, does the laser hit any of these particles? So these vertical rectangles, these are, this is the laser, you can think of it as. So, you know, if we hit a particle, then we're going to get a spectrum from it. If we don't hit a particle, then we're not going to get anything from it. And we're jumping different distances depending on whether we hit a particle or not. That's why these aren't uniform jumps. But, you know, if we do have a case where we're hitting a particle, then what you'll see in the image from the CCD array is something like this. So the rows of this image from the CCD array, that's the vertical dimension along this laser line. And so in this case, you can see that the particle is about 1/3 of the way down the length of the laser line, and that corresponds to this yellow row here. Along the horizontal, that's that wave number. So if you remember that polystyrene spectrum I showed a slide or two ago, it's the x-axis from that. So you can see here this particle. I don't know how well you can see, but there's some speckling here. These are like the little peaks of the spectrum, and then there's also like a big peak out here, which is called the carbon hydrogen stretch or CH stretch. So anyway, this is the data we get from each of these interrogations with the laser. And now just to show you what we do with that, so here on the left is one of those images that has nothing in it, nothing on the tape. And then on the right, this is the example I showed on the previous slide. And if I were to show you like what this one row looks like plotted on a plot, it's this blue curve down on the bottom. So you can see we've got some kind of background fluorescence curve. That's this underlying trend. And on top of it, you've got some peaks, including this carbon-hydrogen bond out here to the right that I mentioned. So this is what we get. In this case, you can see that this particle is maybe like 3 to 4 rows across. Each of these rows of this CCD array are about a micron. So this might be like a 2 to 3 micron diameter particle. And just, I mentioned this earlier, but bacterial cells typically are like 1 to 2. Viruses are like 1/10 of a micron. There's a typo. But that's what we got.
Jared, just so you know, you do have a question from the participants. How come you're using a CNN, not a random forest? So they might be reading your mind forward. Is that something you'll be addressing in a couple slides?
Yeah, I will.
Okay. So
yeah,
that's coming.
Okay. Okay, really quick about how this algorithm works. So we basically go through, if I go back to this picture of the device, maybe. So you'll see that like as we analyze one spot under this laser, we can actually be collecting another spot at the same time. So we kind of like do these things in tandem. So that's what this is. So we'll collect the first spot on the left of that image, then we'll scroll that underneath the laser and we'll analyze that first spot while we collect the second spot. And then we can, shift to them and do the second spot while we collect the third and so on. So when we analyze these spots, what we're typically doing is we're doing a few different exposures at different lengths of time because we want to be efficient with the time we have. We don't want to waste time collecting a spectrum for a spot that has no particle in it. So we do like a quick 1 second exposure just to check if anything is there, looking for that huge carbon hydrogen peak that I mentioned earlier. Then if it seems like there might be something there, by the way, this is what a targeting image looks like over here on the right. So this is an example of a carbon hydrogen peak, this bump out here. If it looks like something's there, then we do a longer exposure for 10 seconds and get what's called a bleaching image. It's called bleaching because also this process kind of, some particles can fluoresce at the beginning as you heat them up with the laser. So typically you might, hit them with the laser a little bit before you actually start collecting a spectrum from them, just to let that fluorescence kind of die down. So that's what this bleaching image is about. And if it still looks like a particle's there, then we collect maybe 4 replicate images at about 20 seconds. So, that's going to be a little over a minute 20 seconds to collect those 4 replicate images. And those are what we use to make the spectrum, which I'll show on the next slide. Also, I forgot to mention, when we initialize the system, we collect a background estimate as well. So that's how it works. And then finally, we have an algorithm to find any particles that appear to be in the image and group those, because you could have multiple particles in one image. So we find the rows for each particle separately, average across these 4 replicate images to improve the signal to noise ratio, remove the background from those images as well to try to remove any of the sorts of background artifacts and things that come from the CCD array or the optical system in general. And then we average across those rows for the particle to finally condense everything down into one spectrum. So this is, in general, how we get a spectrum from particles. And it looks like that. Okay, but you can see now there's this trend underneath the particle, and if you like observe the Rebs collecting these spectra for lots of different particles, you'll see that this background fluorescence level will fluctuate up and down depending on the quality of the particle or the laser interrogation of the particle. So there's still more that has to be done. So improve the quality of the signal, we do this trend removal of this underlying fluorescence. The algorithm we used was called a rolling circle filter. The idea is that you take a circle with a really huge radius and you kind of roll it underneath your spectrum, and you subtract any of the space below the spectrum that these circles cover. And so if you think about a circle with a large radius, it's going to have trouble going up into these peaks, the narrower peaks of the spectrum, but it is going to remove all these like broad underlying trends. So this red curve might be the original spectrum, and this black one is after the rolling circle removal happens. We also have a mapping from the pixel number on the CCD array to the wave number scale, which is actually it's a non-linear mapping. There's this curve is, well, there is a curve here, I guess. This isn't a straight line between pixel and wave number, but that's a minor point. And then there's some other stuff we got to do to clean up. So it's a minor detail, but like each instrument actually has its own mapping from pixels to wave numbers. It's specific to that CCD array that's in that instrument. So to get all these spectra measured on the exact same wave number grid, we use this cubic spline interpolation. And so you can kind of, you can't really see it that much in these plots, but I tried to zoom in on the bottom right here just to show you what's happening. You know, this red curve is the black curve from the previous slide. And what we're doing is interpolating to get this black spectrum, which is going to be on this nominal grid, we call it, which is like the wave number grid we're going to use for all our analysis. So you know, basically we're just trying to remove all of the things that are specific to the instrument or to the you know, collection conditions that led to that fluorescence trend. Trying to get all the spectra on the same scale and on the same grid, so that we can analyze them across instruments.
Garrett, is it possible to get multiple particle types in the same image, like in separate groups of rows?
Yes.
And how do you deal with that?
Yeah, actually we have an algorithm now that tries to go through and do some initial classification. There are like some different kinds of things that can happen in these images, which I didn't really go into detail here, but like for example, sometimes you burn the particle with the laser, and so what you'll see is a spectrum that actually is associated with soot. And in that case, like you can't really recover information about the particle from that. So we try to detect cases when that's happened and sort of kick them out of the analysis process. There are cases when you have lots of fluorescence and you can't really resolve these peaks in the spectrum that you see. So then we kick that out of the analysis as well, but track it to be able to report back to the user if we're seeing unusual levels of fluorescence. And yeah, if you have like multiple particles with signal, you know, in the same image, you know, we have an algorithm that kind of, you can imagine if you look at the at one of these images, right? If you kind of looked along the vertical axis here in a particular column of this array, if you had two particles like right next to each other, you should kind of see like two Gaussian humps or something across those rows looking down vertically associated with each of the particles. So we have an algorithm to try to split particles apart that might be neighboring like that. And, you know, in some cases they're right up next to each other, or particles can clump as well. So you can get some aggregate particles that are just, you know, really big. And we try to deal with that. I mean, we record the size of each particle and try to account for that in the identification if needed. So I hope that helps. Okay. So moving on, because I'm probably taking too long to talk through all this. The next step, finally, the like kind of the final steps, are to take those spectra that are on the same wave number grid now, and we want to remove additional information that is kind of irrelevant. Most of what actually tells you what organism a particle comes from is contained in these smaller peaks in the spectrum, the like narrower peaks. And some of this like broader, these broader features are not as important. So what we do is we run a filter over the spectrum that simultaneously smooths it, but also calculates the second derivative, like a numerical estimate of the second derivative. This filter is called a Savitsky-Golay filter. And so what it does is it basically converts each of these narrower peaks into a V shape. If you think about your second derivative, right, it's going to be positive on the left and the right of this peak, but then negative where the peak turns over. So it'll create this kind of V shape where you have these peaks. And the narrower peaks are going to have a stronger signal in the second derivative space than these broader peaks. And any features you have that are broader than the size of the filter width, which in this case is about 11 values, 11 pixel values, those are going to look basically linear over the size of that filter width, and they'll get zeroed out in the second derivative. So this spectrum is in general like centered around 0, and then you're going to see spikes where you have narrow peaks occurring, which are the things we care about. Also, another detail is that most of the action happens in the smaller region of the spectrum. There's not a lot happening out here in this range. And this carbon hydrogen peak, we mostly use it just to identify when a biological is present. Carbon hydrogen bonds are only going to happen in organic particles. And the shape out here can tell us whether it's a biological versus like polystyrene that I showed earlier. So in general, like once we've used this peak to identify that it's biological, we kind of ignore that in the rest of the analysis. We focus just on this fingerprint region down lower. Okay, so this final spectrum here, we divide by the norm of the vector to make basically like the sum of squares of these values to be the same value. In this case, you can see the scale goes -5 to 5. I think we actually normalize to like so the norm of the vector is the number of values, which is like 380, just so that we don't get like super small values that are hard to deal with numerical precision and everything. But you could make it sum to 1 as well. That would be fine. Okay. So each of those spectra then we need to try to basically collect a bunch of example spectra for different organisms in a library, and then we try to do a classification problem to predict which organism it is. And so, you know, some of the things we've tried in the past, when I originally joined the project, we were doing like principal component analysis to reduce these spectra down into a lower number of components, not to like I'm showing here, but you know, like maybe 10 or 20 or something like that. And then we were using linear and quadratic discriminant analysis to try to predict the organism. Once I joined and machine learning was kind of growing in popularity, we started trying to use like support vector machines, the random forests for those. We tried them on the full spectrum. We also tried it on dimension reduced data. We moved to partial least squares to try to, you know, get like better separation by using supervised methods there. And then more recently, we've moved to like using convolutional neural nets and doing, you know, I think our current method, we don't even do dimension reduction anymore. They work pretty well on the full spectrum, but we have looked into like using things like autoencoders to do dimension reduction as well, or to visualize the library. So this is like kind of a more recent plot of just some of the things in our library, and you can't really see a lot of details, but you can kind of tell how like spectra that come from the same organism, which is the color here, they kind of group together within that space. So I hope that helps answer the questions about the CNN versus random forest. The answer is we did both and compared them.
Jared, some questions are coming in. Do you use non-negative matrix factorization?
I do not know. I don't remember.
Okay. And
Yeah.
then applications of functional data analysis to these chemometric problems are growing. Have you considered functional data analysis methods?
I think there was a time when we might have considered that, and I don't feel like it went very far. I think some of the issues are that I don't like these spectra don't necessarily follow certain functional forms. Maybe it was hard to parameterize things or something, or we just didn't have the budget and the time to look into it. I know we've like discussed some of those things in the past though. At this point, a lot of our algorithms are generally locked down. I think we're not trying to totally change the methods that we use for these sorts of algorithms anymore. Yeah.
So two more questions.
Sure.
What do you mean by library? It's in your autoencoder picture.
Yeah. The organisms that are in the training set, basically. So the classes that you're trying to classify your spectra into.
And did you try k-means? It seems like Kruskal-Wallis is the best fit. How did the convolutional neural network score?
I don't think we've evaluated, I mean, in terms of clustering, we have used k-means a lot for clustering. So we've used like a two-stage k-means clustering where you can like over segment your data and then cluster the centroids of that over segmented set of groups to do some kind of clustering that'll recover large and small groups. Primarily, we've used clustering in the context of cleaning the training data. So, and I'll touch on this in a minute, but we might get like different signatures, different types of spectra in the same set of data, and we've used clustering to try to identify like the good examples of spectra and separate them out from junky spectra or like weird signals that come in into the data set. So it's more used for like outlier removal, I guess, and improve data quality. But yeah, we use k-means too, and it works.
Jared, have you done an evaluation of what methods work best across the ones you've tried through the years? When you moved to newer methods, was it because they were outperforming?
I think once we jumped to SVM, I think SVM gave us a big improvement over discriminant analysis. It could have the problem with like R&D work too is that the instrument keeps changing while you're doing all this stuff, right? So the data quality has improved over time, and also the complexity of the methods we're using has changed over time. So I definitely did see a jump when we went to SVM. Random forest did about the same as that, and I think the convolutional neural network is like a marginal improvement over that. So a lot of it, I think, just comes down to what's easier to use. Also, like when the accuracy of the classification is not that different, it really just comes down to like how flexible are these methods. Something like SVM is a little more finicky than something like a random forest or a neural net. Those you can sort of like throw your data into it and not worry about things as much. Or you don't have to like reconfigure the layout of the model if, for example, the number of values in your spectrum changes. You can kind of handle that more gracefully. In a neural net, you could do zero padding and things to make it work with previous data. So I think that's why we've kind of moved in those directions, is more of the ease of use of the methods. And to deal with just weird artifacts and things in the data, which I'll show in a second.
What do you have to do with your algorithms to put them on the device? Do they just go on as, how do they go on?
Yeah, so our code down at the base level, I guess, we're like running things in C++ or C sharp. What we've done in the past, I mean, primarily our data science team has prototyped these things in R or in Python, and then or in MATLAB. We have done some of that too. And so in some cases, in the past especially, we tried to just like compile that into an executable or some library or something that we could just call in .NET. Over time, as our methods kind of solidified, we tried to rewrite a lot of those things in .NET so that they could just be directly used in the C# code. So a lot of our algorithms do that. And with like the convolutional neural nets and things, we can export those models and use a library to, directly do inference with those models in C++. So, that it does require a lot of working with the software team within their infrastructure, like the software development kind of infrastructure, using, GitHub with version control and everything, trying to make sure that we're following best practices in terms of incorporating our code with theirs. But yeah, that's how it's happened in general.
Thanks.
Yep. Okay, so now I just want to run through like a bunch of different issues we ran into and how we tried to solve them, just to give everybody a flavor of the fun we put ourselves through for the last 15 years. So first of all, the process we use to collect training data is we're gonna, I mentioned this earlier. This is the rebs here, so we inject some target material into this chamber up here. We pull the particles down into the rebs, and we just measure a bunch of these spectra. And because the process we use to collect the spectra, remember, we're just like jumping this laser across the tape and looking for particles. That means that we're not always going to land on a particle. And it means also we're not always going to land dead center on a particle if we do. And so, if we want to establish a spectral signature for a new organism, the first thing we have to do is collect a lot of spectra and figure out which ones of these look like they're from the organism and which ones of them look like background noise. And so, this is like an old readout of what we got from a training run. And I just kind of stacked all the spectra here to show you them all. This is in that second derivative representation, actually across the full spectrum. And this little window out on the right shows that carbon-hydrogen peak in the original dimension, not second derivative, just to help. We used to do this by hand, assign truth to these spectra. Over time, we've developed automated ways of doing it, so because it's really painful. But anyway, here are some examples of what I would call good spectra. So you can see generally clean signal. You can resolve the peaks here, and you see like a strong carbon-hydrogen peak out here, which means that it's like a biological thing. And so these would be cases where the laser kind of landed on the center of the particle and you got nice clean signal. There are some other cases where maybe you hit the edge of a particle or something. I don't even know actually if these ones I called out, we hit anything. This could just be background, but you can see in some cases like this carbon-hydrogen peak is very low. So these would be cases where we hit the edge of something and you might be getting some of the peaks coming through, but maybe the signal isn't as clean. And definitely in these background cases more, it's like really noisy. So the first thing, when I mentioned k-means clustering earlier, we would do like clustering maybe on this set of spectra and try to group them by things that have clean signals, things that have, maybe a partial signature, and then things that are just background. And we'd want to, choose from the good group of spectra. Yes, we can pick up compounds coming from plants. I mean, any kind of biological material we should be able to pick up, as long as it's it's a particle within that size range. So we have seen pollens before, but generally they're larger than what we're going to be analyzing. Okay, so a lot of our early questions were just about dealing with this noise, right? Like you're going to use this device in a real world setting, and you're going to see things where the laser hits the edge of the particle and not just hits the center. So we had a lot of questions about, like, what do we throw in the training set? Do we want to include all of the data that we get for an organism and be representative of what's happening in the real world? Or is too much noisy data going to confuse the model? And so we decided, okay, we'll assign some kind of a noise threshold and put everything that's above that threshold into the training set. And then the question is, well, how do you measure how noisy something is? So we tried a lot of different things. When I originally joined, we were smoothing the spectrum and comparing that to the original, but that only measures the noise. You also have to consider how much signal you get. It's like a signal to noise ratio that kind of matters here. And so then there are a lot of classical measures in spectroscopy for measuring signal to noise, but because of all of this background removal and the rolling circle filter thing that we do, like, this intensity scale loses all meaning by the time we get to the end. So those classical measures ended up not working for us either. And finally, we've kind of signaled on settled on a Fourier analysis approach. So we're looking in Fourier space. The signal is all captured in these higher frequency or lower frequency components on the left, and then the noise is all this higher frequency kind of signal on the right. And so by defining these ranges and taking the ratio of the power in each of these ranges, you can get a signal to noise ratio, which has worked pretty well for us. Another kind of issue that we've run into quite a bit in this is there are different, I haven't really mentioned it yet, but there are like different signatures actually corresponding to the same organism. So as an example, this is an organism. It's a naturally occurring biological. It's called Bacillus thuringiensis kerstaki. It's like I said, it's naturally occurring. You're probably breathing it in right now, but it's a spore-forming bacterium. And so if you hit a spore that was produced from this organism, you'll get the spectrum on the left. But if you hit like a cell from this bacterium, you'll get the signature on the right. And in our training data, in general, it's spores that we care more about. So in our preparation of our samples, when we collect training data, we try to get the bacteria to generate a bunch of spores, but they don't all sporulate. And so sometimes you'll get mixtures of these spectra in the same session's worth of data. And so we need to, again, like, using k-means clustering can help to identify these different groups, but we need to kind of bucket all of our spectra into the different signatures we see. You can tell spores. I mean, I just put a guide up here in case you're curious, but there are some peaks, these blue peaks here, that pop in when you got a spore versus a vegetative cell. And you can also get noisy versions of these. And anyway, so this whole process of truthing, we called it, is actually, when we were doing it manually originally, it was a big deal. Nowadays, we kind of use algorithms to look for some of these peaks. We can use some of our pre-existing models to try to do some like upfront classification into these broad groups of spore and veg, and then do some cleanup on the back end. And it isn't quite as bad. But anyway, we do have different subclasses of organisms. So in the classifier, these subclasses are actually treated as separate classes, and we'll classify into one or the other for a test spectrum. Another general issue that we've run into is in our case with this classification problem, for organisms that are similar to each other, Sometimes the between class variability can be less than the within class variability, which makes classification difficult, of course. Just to try to illustrate this for you, I took 10 spectra from the same subclass and plotted them on top of each other on the top, so you can sort of see the spread that you get across different spectra from the same thing, same organism. And then down below, these are 5 different organisms. I just took the average spectrum across all the training data we have for those 5 organisms and plotted on top of each other. And I mean, whoops, sorry. You can see places where these signatures might deviate from each other a little, but in general, like the spread you see across these 5 organisms isn't that different from the spread you see across these 10 examples from the same organism. So, that means we've got large clouds of data from each organism, if you think about an n-dimensional cloud, but then also those clouds are sort of overlapping on top of each other a lot too. So that can cause problems. We do try to decrease the within class variability, at least in our training set, by using signal to noise filters, try to detect and remove outliers, just really weird artifacts and things we get in the data, or try to understand where those artifacts are coming from to see if we need a different subclass. And then try to help use more complex models in some cases to key on whatever differences are here, and improve the accuracy a little bit. And then another thing we have done more recently is try to classify things at different levels of specificity. So sometimes it's easy to tell that something's biological or that it's a spore, or even that like this is a bacterium versus a virus or something. So you can return results at that level at a higher confidence level, and then maybe make a call at the organism level that's less confident. But sometimes this high level stuff can be helpful for downstream analytics. Another thing about subgroups, or rather a thing that just makes the problem harder, is there are definitely like instrument level features that creep into the spectrum, and there are also features associated with other environmental factors, maybe the humidity, the temperature, the time of day. These things all, it's challenging when you're building a system for the field to account for all these sources of variability and to even measure them. But definitely we observe them. So this just as an example, this is a partial least squares plot in 2 dimensions, just to kind of show you the data. This red cloud is this one of these Bacillus thuringiensis kerstaki, or BTK for short. These are a bunch of spectra we collected on that organism. And if you take this red cloud and you recolor it by like the session, so a session is like a single run of the instrument. So when we disseminate BTK in this case, and we like run the machine through one cycle of collecting a spot and then jumping across that spot and collecting all the different particles in that spot, that would be a session. So, sorry. So when I break that out by session, you can see here that some of the sessions, like this brown one here, kind of clump up in this space. And also, if we collect on multiple instruments, you can see that the instruments kind of break out into clusters too. So there are definitely sources of variability associated with the session, which is the environmental condition, and the instrument that you can see through dimension reduction techniques like this. So a lot of times we've made plots like these in a diagnostic setting just to try to identify potential issues or other things we need to track and include in models potentially to mitigate their effects. Okay, another issue that we run across is just weird stuff with the hardware, kind of, or with the collection itself. So this rolling circle filter sometimes gets messed up by fluorescence signatures. This huge spike in the spectrum at the beginning here. This is called Rayleigh scattering. It's the light coming back from the laser at the same wavelength that the laser is. So the light that didn't get shifted, basically, and there's typically a ton of it. So we ignore like below kind of where this arrow is pointing. We ignore that in the analysis, but when we do this rolling circle filter, if we catch some of this front part of the spectrum here, then the circle doesn't quite fit into that curve there from the fluorescence, and that can end up pushing up the beginning of the spectrum. You'll see like a slope in the spectrum at the beginning. So sometimes the rolling circle will fail like that, and typically we have to like tighten the analysis range. This is sort of an issue that we revisit every new generation of instrument, because sometimes the optical system will change a little bit, but it's a thing that happens and we just learn to recognize it. Another thing that happens is sometimes the pixels in the CCD array will have like a different intensity compared to their neighbors. So this is a background image from a CCD array, and you can see some of the pixels in it just look brighter. So if you have a pixel that's like more sensitive to light than its neighbors, and that's in the background image that you have, then if you remove this background signal that has kind of a spike at this one point, you'll end up creating like an inverse spike, like a dip in your spectrum. And so you'll see a dip in the spectrum like this, which is not good. But then also when you do this rolling circle and you hit a dip, a big dip in your spectrum, that circle can't like fit onto either side of the dip, and it ends up like pushing everything up in the spectrum. So you can see this kind of like general trend here. That's just from the rolling circle, like pushing the spectrum up because it couldn't fit up into here because of this dip. And it creates like a kind of volcano shape. So things like that happen, and we learned to detect those and try to mitigate them as much as we can. Other things happen like this etaloning pattern, it's called. It's a thing that happens with CCD arrays where you get sort of a wavy pattern in the image, and the wavy pattern is not necessarily consistent across time. So when we've seen this kind of thing, especially with when we have low signal in the data, it creates this sinusoid pattern, which is an issue. So we've had to try different things to try to mitigate the effects of this kind of thing happening too. But these are the fun things that happen when you're dealing with real data as well. And then just a quick note on how we tried to identify or mitigate these things. I mean, with these bad pixels or the different sensitive pixels, we just kind of smooth over them. We detect them and we smooth over them, which has helped, especially in the background. Adjusting the analysis region for this, like I talked about. And then the background removal helps a little bit with this etaloning. We've also done some stuff in Fourier space to try to detect it and remove it. Don't have a lot more detail there to share. And then all these things too, better hardware does help over time. So some of these things have just reduced in, like the impacts have been reduced over time because the hardware has gotten better. Yeah, I'm almost done here. Just a few more slides. So one big thing on the algorithm side that we've continually struggled with, and I think most people who do classification struggle with this as well, is that our classifier is trained on a set of known organisms. So organisms we decided like these are important things to detect with the REBs. We went through the process of disseminating them, collecting the spectra, and building like a training class for that organism. And that forms our spectral library. So the library of spectra and the labels that go with them for these organisms we picked. The problem, of course, is that in the field, if you stick this sensor out in the middle of a park somewhere, you're gonna see lots of particles from things that aren't in that library, organisms we never collected. So the problem is, like as I showed up here in the top right, I mean, if you have a model that's trained to look at images of animals and tell you what type of animal it is, and you give it this picture of a garden hose, it's going to say that looks like a snake because all it knows is about pictures of animals. And so similarly with our classifier, when we've got like this specific universe of organisms we're looking at, and we get something from a new organism come in, it's going to predict it as one of those organisms, or maybe it's going to not. It's going to give you a prediction that's like spreads the probability out across lots of groups, which is what you would hope would happen. But in practice, it doesn't always happen that way. So sometimes we'll get a high confidence hit on the wrong organism. So one big area of research for us has just been like, how do you detect when this happens so that you can avoid misclassifying a particle as something? And so, we've tried a bunch of different approaches. You can do like one class kind of models where you sort of try to draw a boundary outside of your training library. And if anything's outside that boundary, then it's predicted as out of library. We've tried doing things like that autoencoder I showed earlier to sort of describe the entire library and try to see how close a new spectrum is to the existing classes. And we've also, like I said earlier, how we have that multi-stage classification. Incorporating things like that can help reduce misclassifications as well, especially when you pair it with some like decision tree kind of logic. But this is an issue, a big issue in classification. Okay, and then just a hodgepodge of other things that came to my mind thinking about this work. In general, when you're doing research and development work, it can be hard because you're kind of developing algorithms for a moving target. The hardware underneath everything keeps changing, so that means the data quality keeps changing over time, and the same kinds of machine artifacts that appeared in old data now appear in different form in new data. There's also like the whole contract research side of things where or the the internal R&D kind of ebb and flow as well across fiscal years that makes it like hard. Sometimes you can only do algorithm work at certain times of year or every other year or something. And then also as the market changes, we have to keep pivoting this device toward those new markets. Like I mentioned earlier, how we've focused a lot on shrinking the size of the instrument, which has caused problems sometimes with the data quality. So the other fun thing on this one is, if you think about it, collecting data on a really dangerous organism like anthrax or something, that's not like something that you can just do easily. It costs a lot of money to collect these spectra in a high containment environment. And also you want to limit the exposure of your instruments to, seriously bad pathogens like that. So for those types of organisms, we typically like only get a couple days of time with one instrument in the chamber, and then all the data in your library from that organism was basically collected on one instrument at one time. So it makes it hard in the future to make sure that training library entry is robust. Say.
Here, one more question from the audience, and that's an ask if any small organics from, say, trees have been tested in these kinds of instruments. So I think the crowd's interested in, other uses for these devices.
Yeah. I mean, again, we're focusing on particles small enough for you to breathe in. So, I mean, that's the main use case for these. So a lot of the things produced by plants and trees and stuff are not going to be in that range. That said, I mean, we've done some side projects to explore using this technology in other circumstances. So we built a liquid sample adapter that let us take samples of water that maybe we want to monitor for fungus and mold, and that can be pulled into the REBs and sort of dried out within that liquid sample adapter and then presented to the substrate to go through the regular process. So we can monitor liquid streams like that. Again, though, looking for things in that size range. And then we've also done some chemical analysis as well, where we had to build a sort of a different, the sample collection and presentation to the laser had to be a bit different for chemicals, but we've done that as well. So the REBS has worked in a context of detecting and identifying chemical mixtures. But yeah, I don't think we've looked. I don't think we've found like a market that wants us to look at trees yet, and so we haven't. But I think, yeah, depending on the need, it could probably be done for sure. Okay, so conclusions. Okay, just some general lessons that I've learned working on all this stuff. A lot of the issues that I talked about earlier resulted from somebody just looking at the data. And sometimes when you're analyzing a bunch of observations and stuff, you don't necessarily have the time to look at just the raw data or the process data at different points in the process to like make sure things are still working the way you thought they did. It's easy to kind of like after doing that initial R&D work to set up the algorithms, it's sometimes you fall into a habit of not looking at the data as much anymore. But definitely, I think that's something we all try to remember to do because you can see some really weird things happening sometimes when you look at the data, and then you need to figure out what's going on there. I think understanding where the variability comes in your data is also really important. I think it's good at the beginning of projects. It would be, I would like to see us do that better, you know, to just kind of think about all the different sources of variability and think about ways that you might be able to measure that in some way so that you can account for it later on in the process. One thing that we've seen happening, so I mentioned earlier how each session of data, like they kind of break out into groups on their own. We found when we were early on doing the modeling, we were just kind of splitting our spectra into a train and test portion randomly, like an 80-20 split, without realizing that there was so much similarity between spectra from the same sessions. And after, like, what that ended up doing then is our classifier results were optimistic in terms of accuracy. It looked like they were working really well when they actually weren't. There was some kind of not a direct leakage between train and test, but like a leakage through the correlation of the data that were in the same sessions. So once we realized that, we started holding out entire sessions as train or test. And then we were getting better estimates of the accuracy of our algorithms. So, you know, definitely think about sources of variability and think about sources of commonality in your data also. You know, where is correlation in there, and try to make sure you account for that when you do training and validation and testing of your models. The other thing, I mean, we would always laugh, like if we did anything on this project after a while and it seemed like it worked the first try, we would all just start being like, okay, something's wrong here, because you know, basically, like most of the time when something did seem to work, it actually hadn't, and we had done, you know, something stupid, or that there was something happening with the data that we hadn't appreciated that made things look better than they were. So definitely when you're in R&D mode, especially, like I would say approach everything with skepticism, and also like you know, if you think that the method you're doing is the best, have the, you know, humility, I guess, to consider that maybe it's not, because a lot of times there are better ways of doing things. So that was something I appreciated about our team, is we were good about you know, being curious and open-minded about new ideas.
Here, there's another question from the Q&A, and that's about the concept of using Kruskal-Wallis to create since you got a frequency distribution on a matrix from the laser, the Kruskal-Wallis could convert that and cut out the CNN, so you could eliminate the camera. You're creating a frequency distribution on a matrix with a laser. It seems that Kruskal-Wallis is the most definitive. Kruskal-Wallis is way more computationally efficient. Cutting out the convolutional neural network will eliminate the camera and data quality issues. think they're,
That I would have to think about. I don't know. Let me jot that down and think about it. Okay.
is it okay to share your email in the chat in case this person would like to reach out to you?
Yeah, sure.
Okay, I will do that. Thank you for that question. It's always good. It's great to get recommendations.
Yep. Absolutely. Okay. Yeah, and I think this is the end here. So yeah, getting a second opinion is always important, I think. We always had a lot more success when multiple people thought about the problem to identify potential advantages and disadvantages of the different solutions we had. And like I kind of have been alluding to, the team dynamics I think are really important. We do a great job at Battelle, I think, of having interdisciplinary teams who are all kind of team focused, working together toward solving a problem from different kind of orthogonal perspectives. And all of that works really nicely on a project like this, where there are so many unknowns and so many challenges to overcome. So I think that brings me to the end of the talk. Oh, I'm off my slides here. Yeah, okay. I thought maybe I stuck my email in here, but I didn't. But yeah, it should be in the chat then for you to see. And I'll stop there unless there are other questions.
Thank you so much, Jared. That was a very interesting presentation. It was good to hear about the science and what you're doing with the data. No more questions in the chat, so appreciate your time today. And this recording will be available on NISS's YouTube channel, and Jared's offered to share his slides in a PDF format. So we'll make those available through NISS too. Thanks everybody for attending today, and we'll see you next month.
All right, thank you.
Thank you so much, Jared.